Okay, I'm going to turn it over to Dr. Morton for a few minutes, but first I want to say something about the exam, and then he's going to talk about what you're doing in lab this week besides the lab quiz, and then I will be around if you have questions about the lab quiz toward the end of the period. I've got one more makeup to give, and so I can't give your exams back until 1:30 today. You can pick them up at Natalie's office after 1:30, okay? Much better performance on this exam. I don't know if you remember, but the mean for the class last exam was 68. It's 79 this time. And here is our grade distribution. So a lot of people up into the 90 and 80 ranges, which is nice to see. And so much better performance on this exam. High grade was 104 out of 100. And so somebody really knocked the socks off of it. One other quick thing, announcement. There's another optional point opportunity for you for those that want to do optional points, 20 optional points. There is a guest speaker, Matt Rims, graduated 13, and he's going to present on his master's work in statistical genetic epidemiology at 4:30 in Mars 208. He was a math major, got his biostatistics master's at Ohio State University. And if you have any interest in human health, Matt's talk should be relevant and interesting. So, again, tomorrow at 4:30. So, I'm going to turn it over to you, Larry, and then we'll...
All right, in lab this week, we'll start off with the lab test, and it probably will not take more than 30 minutes if you understand the material reasonably well. Some of the questions that came up last night at the review session, it's not formatted like your regular classroom tests, right? In the sense that it's not a lot of multiple choice questions, although there are some multiple choice. Most of it is short answer, single word, analyzing data, right? And and describing, how you came to a conclusion based upon your analysis of the data. Okay? If you go over the study guide and do the things that are on the study guide, you shouldn't have any trouble. Okay? Please be on time. I will start the test at the beginning of the lab period, whether you're there or not. Right? I won't start early, but I'll start at the beginning. There is a lab booklet that we will use for each of the periods. Please do not write in the lab booklet. You will have an answer sheet that you will fill in. Okay? But other than that, that's what the first half hour will be.
All right, just a lot of the questions are similar to the kind of questions you answered on your lab handouts. Okay, so those types of questions are the kind of things we're looking for. Can you analyze your data? Can you
It's a lot of
look at some data and see if there's statistically significant differences and draw conclusions based on that?
Be sure you understand how to use StatBook. You enter data on the spreadsheet, you get numbers at the top, mean, standard deviation, and you push the T-calc, you get a period of numbers down at the bottom. What do they mean? How do you use them? Okay, if that has been a challenge for you, be sure before you come into your lab class that you understand how to do that, how to use that to determine whether two data sets are statistically significantly different. Okay? All right. You have two challenges left for Lab 5. One is a thing we refer to as data transformations, right? Generally speaking, you do not present raw data when you present the results of your experiments, whether it be in an oral form, poster form, or a publication form. So we've got to do something with that data, right? And this is what we refer to as data transformations. We're going to take the raw data and we're going to reduce all those replicates you did, 2, 3, 4 replicates, to averages and do some statistical analysis on that. Now, for most of you, your data might look something like this. Lots and lots of lines. And if you did as recommended, you went in and you changed the name of the lines from run 1, run 2, run 3, to whatever the particular variable was for that set of runs. So we have to take all this confusion and make it understandable. Sift out the triplicates that you have for a particular run. Okay, and then take that data and average it for each time point over the 180 seconds. If you were really lucky, your raw data might look like this, where your triplicates are extraordinarily tight. But for the most part, this didn't happen. Right? When you follow the protocol in your lab manual, we will be using Logger Pro. What you will generate would be a graph that might look something like this, where each of these lines represents an average of triplicates. In this case, for pH, there's a legend up here. So each of these represents the enzyme activity for one pH, 2 other pHs, and so on. Right? This is far more understandable and far more interpretable than your raw data. So the big challenge today, or one of the big challenges today, is to follow a fairly complex but orderly protocol that's in your lab manual for taking your raw data and averaging your triplicates so that you can generate a graph that's far more interpretable. Okay? Then the question becomes, what does this tell me? What does this tell me about the activity of the enzyme under a particular condition? Some of you did temperature, some of you did pH, glycol concentration, peroxide concentration, both, or some of you used an inhibitor. How do we describe the efficiency of the enzyme under each of those different conditions? We're going to use a utility that's built into Logger Pro that will allow us to take a section of that data and determine the rate of the chemical reaction. So what is a rate? What would be the rate of the enzyme reaction? What is the denominator in a rate reaction all the time? Time. All right? What's going to be our numerator if we're using that data? Absorbance. So we're going to be looking at change in absorbance at 470 nanometers with change in time. In a graph like that, what aspect of that graph represents this value? It's going to be the slope. Okay? So what you're going to do is select the time period. You're going to select the time period for these graphs, and it has to be the same time period for each data set, where the slope of the line is as close to being linear as possible, and also is as early in the assay as possible. So for the most part, the best data most linear data is going to be here in the beginning. This is where your substrate concentration will not be limiting in any way. You will not have run out of glycol or peroxide. That's why these graphs tend to decrease in slope as you get further and further out, right? Because you begin to run out of substrate up here. So we're going to stay away from these points here and look at these points down here where the data is fairly linear. All right? So with LagerPro, you're going to be able to select a period of time, and I'm just arbitrarily doing this, where the data for each of the experimental variables is fairly linear. And up at the top of the LagerPro screen is a button, an icon that you can push that has a thing that looks like this, a line and an R. That built-in utility will automatically determine the best fit line for each of these graphs and give you a bunch of information. It'll tell you the y-intercept, it'll tell you a variety of things, but what we are most interested in the data that comes off of that is M, the slope. The slope will be the change in absorbance over change in time. Now the change in time will be in seconds, right? Okay. The slope then will be a reflection of the rate at which that enzyme is able to function at each of these particular variables. In this case, we're talking about pH, but if you do temperature or glycol concentration or any of the other variables, you will be able to compare the enzyme activity here to here to here and so on. And you'll be able to do it objectively because you will have the slope of those lines rather than just to look at it and say, well, it's obvious that this pH works better than this. Yeah, but that doesn't work in science. In science, you need to do some type of objective analysis, in this case a statistical analysis, for us to say that there is a difference. Okay, so what if you wanted to actually do a t-test to find out if these are indeed statistically significantly different? The averages alone won't tell you that. Right? You'll have to go back to your raw data. Right? And for a time point within this range, you can pick, let's say I take the 50-second time point, and I say at 50 seconds, are these values values of the enzyme activity at that particular time statistically significantly different. So you can go back to the 50 seconds, take the three values for each of the pHs you tested, enter them in the stat book, and you'll be able to do your comparisons using the t-test. Okay.
And there is a utility on LagerPro that will allow you to put your cursor over those lines, and it will then show you at that time that particular value, that absorbance value.
What Dr. Browder is talking about is when you move the cursor into the graph field, you get a thing that looks like this. If you put that over a certain point in the graph, down over here, there's a little box that gives you the X and Y values. So if you select 50 seconds, right, one of the numbers down here will be the X value, 50. The other will be the Y value at that particular time point. So you can find the exact value at a particular time point for each of the replicates that you do. This way, then, when you do your presentation, you will be able to say that the enzyme worked far better at pH 6 than it did at pH 5, with a 95% confidence value. Right? You'll be able to do that in your presentation. Rachel?
How many time periods do you want us to do this?
You just do it in one. And you pick one within that same time frame that you did the best fit one. Okay?
But make sure you do the same time for all of them.
Correct.
Now, realize this, you're only going to be entering 3 values in the stat book. So your degrees of freedom is only going to be n minus 1 plus n minus 1. That's 4. And if you look at the t-table, and degrees of freedom 4, that t-value is fairly high. So you've got a pretty big difference in those means for that difference to be significant. That's why if you do more replicates, it gets much easier to determine if the difference is significant. The threshold's a little bit lower.
This is a bit of a step up from what you did last semester in 134, when you had to present the results of the little experiment we did at the end of the semester. Right? But as you proceed through your years here, this type of presentation, an oral presentation or a poster presentation, will be something that you will get increasingly better at by the time you finish.
One of the reasons we set up this little experiment is this is going to mimic something that happens in grad school a lot, called a lab meeting. And in lab meetings, different parts and different groups are working on different parts of a bigger question, like you were. Some of you picked pH, some of you picked temperature. But then everybody gets back together again to share their data, because you can learn things about your experiments by looking at other people's experiments. And so we're trying to kind of give you a first introduction to mimicking this data sharing. And it's not just grad school anymore. It's jobs, it's everything. There's a lot of collaboration that takes place, and one group's got to let another group know what they're doing so that you can all get a bigger picture of what the bigger question is.
The left hand has to know what the right hand's doing if you want to make any progress. Okay, so that will be the main focus of today, to get through those data transformations and analysis, right? But then you've got to put it together in a PowerPoint presentation. You can use Keynote if you're a Mac person. That's perfectly fine. And I guess there's some other types of presentation software out there I'm not all that familiar with. But either of those will work fine. Okay? So I'm going to give you some pointers about doing a PowerPoint presentation, because it's easy to do it poorly and difficult to do it well. All right? You have a story to tell. Last week when you came into lab, right, you had a goal, right? You had a process that you went through. The PowerPoint presentation is there to help you tell your story. It is not the story. You are going to tell the story, but let the PowerPoint presentation help you tell your story.
Larry,
Yes.
can we email these to them so they don't have to try to write down all these slides? Can we just email this back?
I'll email it to you.
Okay,
Yeah.
I'll send you the PowerPoint. So.
Right? Let the PowerPoint slides help you make the points that you want to make. Okay? And we'll talk about how to do that next. Be sure it's easy on your eye, easy to read. Look at this. The background that I have here is very simple. It's one of the standard backgrounds that you can find in PowerPoint. Right? It should be easy to read and not cluttered. I know kids kind of like to have, lots of eye-popping stuff. As long as it doesn't detract from the presentation, that's fine. You'll be including text and images, and you want it to be something that the people will remember when you're finished. They'll remember your conclusions. They'll remember how your data supported the conclusions. Okay? The components of the presentation will include some type of review of background information. What have other people done? Or why did we do this? And it's not because it's the next lab in the series, no. What was it about this particular exercise that was an important goal for not just your group, but the entire lab group? Okay? What was the question or hypothesis that you were testing? If you did pH, what was the optimal pH for, ternary peroxidase activity, or temperature, or what effect did hydroxylamine have on the activity of the enzyme, and was it a competitive or non-competitive inhibitor? And which substrate was it competing for? All right? The biggest mistake that kids often make is with the methodology. You don't just list a bunch of steps. Okay? You can assume that everybody in your lab period knows the basic protocol for running the exercise. What you need to do is emphasize what was your variable and how did you change it. Okay? Sometimes a flow chart works really, really well. Picture's worth 1,000 words. It really is. You can get a whole lot across with good visual representation of what you did, rather than a bunch of words on the text. Right? When you present your results, you describe them only. You do not draw conclusions at that point. Conclusions come in the next part. Right? But when you describe the results, it's really important that you emphasize the aspect of the results that is either consistent with and or not consistent with your hypothesis or your question. What contributes to you addressing your question? Remember, this is a story you're telling. You want to take the people in the room figuratively by the hand and lead them on a path through your story. Help them understand what it is you did, why you did it, what you got, and what the conclusions are that you can draw based on that data. All right, with respect to text, keep your font simple. All right? I recommend a font like Arial or what's the other one? Helvetica, right? That's a sans serif font. It doesn't have those little feet, although Times Roman is fine. All right, stay away from funny things like this. Okay. Use bullet points. If you're going to list something, bullet points help you separate what it is you're going to say into definable pieces of information. All right. Use keywords rather than sentences. The worst thing you can do in a presentation is to read sentences off the slide. Right? You're going to be presenting to your classmates. It's not like it's going to be a vicious group of people looking to make you look stupid, so you don't need to be nervous. But let the slides prompt what you're going to say. Don't have the slides say everything you want to say. Right? Keep it simple. No more than 7 lines of text, no more than 7 words per line. That's a rule of thumb I learned decades ago. I don't know if that's still holds true. Be careful of the font, not only the font you use, but the color. You want there to be good contrast. Clearly, you can read this white on blue very easily. Sometimes there's a tendency to want things to pop out of color. And one of the biggest mistakes is to use a yellow font on a white background. You can't read that. So be careful about the font, not only the font, but the color of the font that you use. Many of the pre-designed backgrounds that are in PowerPoint pre-select the type of font and the color of the font to avoid these sorts of things, but that's not to say you can't be a little creative. This is a typical mistake that students often make. They'll stand here and they'll read this. All right? You do not want to do that. There's lots of good information here, but if it was in a series of simple bullet points with simple catchphrases, you get a lot more out of it than try to sit there and read it. You will lose your audience if you expect them to read that while you're talking to them. If you read it to them, you're going to lose your audience. They don't want to listen to you babble on what's on the board, right? Here's an example of the use of bullet points, although I would not recommend that you necessarily do this by describing what's in 2, 1, 2, and 3, because everybody did the same thing except they had one thing that they varied. It's what they varied that's important, not necessarily what's in each tube. All right. Graphs and tables. You will have graphic data as well as tabular data. Right? Be sure when you present your data that you describe the axes and the units, if there are any units. Absorbance doesn't have any units, but you want to be sure that the people in the audience understand what type of information is contained in the graph. Right? If you have a legend, that's good, as long as it can be read. You can use text boxes to particularly identify the lines on a graph. Make it easy for your audience to follow your description of the data by giving them visual cues that support what you are saying orally. When you describe the data, be sure to present it in such a way that it addresses the question or hypothesis that you are testing. For example, if you were looking at pH, hold on. If you were looking at pH, you would want to describe this data in such a way as to emphasize, you're only going to describe it though, that this particular pH, the enzyme worked much more rapidly than at this pH. You can still be descriptive without being interpretive. Right? Help yourself, help that group that you're leading by the hand through your presentation to come to the conclusion that you believe your data supports. Right? If they have questions about it, if after your presentation somebody in the lab group says, yeah, but what about, right? They'll ask that. They'll have the opportunity to ask that at the end. Lead them through your logic. Lead them through your story. This is the sort of thing you want to avoid. What is this? There's no title, right? It says control, what? Experiment 1, what was the variable in that experiment? Right? You have numbers here, but there's nothing descriptive as to what those numbers represent. Right? There's DOF and this. Okay? This data can be presented, but presented in a way, as I mentioned, where there are descriptive titles that help the individual understand what it is this data represents. Don't just throw stuff on the slide and expect them to understand it. You have to explain it to them. Okay? If you use pictures, and this is perfectly fine. If you download something from the internet, and believe me, there's a lot out there on the internet having to do with this particular exercise or something very similar. It's a fairly widely used experimental design to look at the effect of variables, environmental variables on enzyme activity. As I mentioned to you, preparing tuna peroxidase is like mindless. It's so easy, so it's widely used. Right? If you download images from the internet, be sure you give an attribution to those images. Where did you get it from? What was the URL? Right? It's far better to use a JPEG than a bitmap. I'll show you why in a minute. Right? Because pixel density should be high, so the picture stays really clear. Don't overly enlarge, because then you get pixelation. And don't change the aspect, and I'll tell you what I mean by that in a second. So here's a nice picture of a flower. It's a JPEG. It's nice and clear. This is a bitmap of a rose. And you can see the bitmap doesn't provide you with the same pixel density, the same clarity as does a JPEG. If you change the aspect, you can change the way relative proportions of the image. So here's an image of E like we did in the microscopy lab, right? If you simply drag one axis, say, or one edge, this edge, pulled in this direction, you elongate only that dimension. If you want to enlarge, be sure you pull at a corner, and that will enlarge both X and Y proportionally. Now, that may or may not affect the quality of the image. I know oftentimes you'll see in student newspaper publications, they have a picture that they want to put in the article, but it doesn't quite fill the space, so they just drag it to fill the space, and then your face gets wide or it gets long. What about backgrounds? Be sure they don't distract. You don't want a cluttered background. You want the audience to focus on what you're saying and how what's on the slide contributes to what you're saying. It supports it. Right? Be sure you've got a good contrast between the background and the text. Right? I'm not sure if I still hold any of these. I would suggest a darker background and a lighter text rather than a lighter background and a darker text, but it varies on how you want to present. Animations, right? This can contribute to making points individual at a time. If you have a bulleted list, sometimes the animations can actually contribute, but they can also distract. So be careful of how you use animations. Keynote and Mac-based presentation software tend to be heavier on animations, things you can do simply because the architecture of the computers contributes to that. But be careful that visual and auditory things, like you can set this up so as something comes in and you hear there's a sound. Right? Again, be sure it doesn't distract from you making your point. As I mentioned, the animations sometimes help you focus the audience on a particular point. Rather than putting all the points up at once, you can put them up one at a time and keep them focused on each of the new points without getting them distracted by the other points. Right? You can have transitions from slide to slide, which sometimes are kind of nice. Finally, in conclusions, lastly, be sure it is absolutely essential that the conclusions you draw are supported by the data and the data analysis that you have done. You cannot draw conclusions that are not consistent with your results. Okay? You can present a summary of findings. It can be a bulleted list. What is the meaning? What's the significance? Why do we care if enzymes activity is affected by temperature or pH or inhibitor? What is the significance biologically to that type of control? Right? And you will, it's always good to see how what you did compares to what others have done. Now, is it on Cell Bio Resources with some previous data? Yeah. When we opened up LivePro, there was that menu that had cell biology resources.
No, not longer. It's on the desktop.
It's on the desktop?
Yeah.
Okay, so it's an icon on the desktop where the results of students from previous years are posted, and you can compare your stuff to theirs. Right? It's always important when doing science to compare the results that you got to what has previously been reported. It may be consistent, it may not, but this is one of the great things about science. It's self-correcting. If somebody comes up with new information that's not consistent with what has been reported in the past, scientists stop and go, oh, do we need to change our idea of what's going on here if we're going to consider this new information? So take your data and compare it to previous students. You can also go online, and in many cases, other schools, colleges, universities, even high schools have posted the results that other students have gotten for this or a very similar experiment. Right? That's perfectly valid. Any questions? Okay, so when you come to lab this week, beginning today, you will first start off with your lab test, and then you will have the rest of the period to work on your data